Hi, my name is Kitty Huang. Today is Sunday, January 23rd, 2022, and this is the video for Case Connection Week 2. So I want to start off by introducing you guys to Bob. He is a 16-year-old male who has exercise intolerance, muscle fatigue, and dark urine. He has had difficulty with exercise and would often feel fatigue and muscle pain with any strenuous exercise. Initially, he and his parents thought his symptoms were due to him being a little bit overweight and chalked it up to growing pains. However, Bob has noticed recently that whenever he would exert himself, the pains were severe and was followed by dark urine. His pediatrician ordered blood tests, DNA tests, and muscle biopsies and confirmed that Bob has a deficiency of muscle glycogen phosphorylase. So how is muscle glycogen normally used during exercise? When we eat carbs, our bodies break it down and changes it to a form of sugar called glucose. Glucose then goes through a series of processes that turn it into glycogen. Glycogen is the predominant form of sugar that is stored mostly in our liver and muscles. Glycogen is very important because it is what our cells use to produce ATP for fuel and energy. Glucose is delivered into muscle cells by transporters called GLUT4. Once in the muscle cells, glucose is converted into glycogen for storage. Muscle glycogen is unique because its storage is available for use within the muscles itself. During workouts, when the muscles need a supply of ATP, a molecule called adenosine monophosphate, aka AMP, starts to build up and signals to the muscle cells that it needs to produce more ATP. This signaling activates an enzyme called glucagon phosphorylase, which releases a glucose enzyme called glucose 1-phosphate. Glucose 1-phosphate then becomes glucose 6-phosphate and ultimately glucose. This available glucose goes through a process called glycolysis, which turns it into pyruvate, then acetyl-CoA, and ultimately ATP. So, I know Bob likes long walks, and his concern was whether he can still do that. He has a rare inherited disorder called McArdle's disease, aka glycogen storage disease type 5, and it mainly affects skeletal muscles. McArdle's disease results from mutations of the glycogen phosphorylase enzyme, also known as PYGM. Because PYGM is responsible for releasing glucose 1-phosphate, this deficiency will result in the inability of his muscles to produce ATP from glycogen. Symptoms are commonly exercise intolerance, muscle cramps, weakness, fatigue, and damage. However, evidence has demonstrated that eating a high-carb meal or snack prior to physical activity prior to a moderately intense aerobic workout actually reduces these symptoms. Physical activity should be done in a slow, gentle manner and slowed down. Once restarted, he may experience something called the second wind phenomenon, in which the initial pain and high heart rate will decrease and his tolerance will improve. This happens because of increased blood flow to the muscles and its ability to find alternative sources of energy like glycogen from the liver. Does Bob need to eat at night to maintain his blood glucose levels? McArdle's disease affects the muscle's ability to produce ATP. This does not affect his blood glucose level. His liver is responsible for maintaining blood glucose, so therefore he does not need to eat in the middle of the night. However, he should still maintain a high-carb diet throughout the day because his body will use this carb in his liver to produce ATP by breaking down what he eats. Does Bob have to worry about producing excessive lactate during intense anaerobic exercise? In the cells, glucose can be broken down to either pyruvate or lactate depending on the availability of O2. These two pathways require glycogen phosphorylase Because his glycogen phosphorylase is ineffective, he will not produce lactate. Lactate is a backup source of energy when his muscles have depleted their stored glycogen. This is the reason why he is unable to tolerate strenuous exercise. Unfortunately for Bob, while he was getting worked up for his symptoms of fatigue, muscle pain, and dark urine, his pediatrician also noticed that his biomarker for PKM2 is unusually high. Prior to this, Bob has never even heard of pyruvate kinase. He is told that pyruvate kinase catalyzes the rate-limiting ATP-generating step of glycolysis, in which phosphoenolpyruvate, PEP, is converted to pyruvate. Multiple isoenzymes of pyruvate kinase exists in mammals. Type L, which is found in the liver and kidneys, type R, which is expressed in erythrocytes, type M1, which is found in tissues such as muscles and brain, and type M2, which is present in the self-renewing cells such as embryonic and adult stem cells. So how is pyruvate kinase regulated in the cells? Glycolysis is a series of steps in your cells in which glucose, a form of sugar, is converted to pyruvate and eventually acetyl-CoA and ATP production. Pyruvate kinase is an enzyme that is involved in the last step. Under normal conditions, pyruvate kinase is regulated three ways: allosteric effectors, covalent modifiers, and hormonal control. Allosteric regulation happens when an enzyme, in this case pyruvate kinase, is bound by an effector molecule, causing a conformational change within the enzyme. Allosteric regulators can be activators or inhibitors, and they are very fast-acting. In the case of pyruvate kinase, its allosteric activator is fructose 1-biphosphate, and its allosteric inhibitor is ATP and alanine. Hormone inhibitors are glucagon, R-glucagon, cyclic AMP, and epinephrine, whereas hormone activator is insulin. Covalent modification is a slower process because it changes the protein structure. Pyruvate kinase is deactivated when protein kinase A attaches a phosphate to it through a process called phosphorylation. High levels of insulin, on the other hand, activates phosphoprotein phosphatase 1 and increases the action of pyruvate kinase. Many types of cancer cells are predominantly glycolytic and overexpress the oncoprotein MYC. Interestingly, the classical oncoprotein MYC has been found to promote preferential expression of pyruvate kinase type M2 over pyruvate kinase type M1. The isoform M2 is characteristically found in a low activity state and is inactive at promoting glycolysis when compared to pyruvate kinase type L or M1. This logic seems counterintuitive, and for a glycolic tissue, what is the rationale for this mechanism, and what other pathways may cancer cells be diverting substrates to? Pyruvate kinase muscle isozyme M1, aka PKM1, is non-allosteric and expressed in different tissues which require a large supply of ATP, like the brain, muscle, and heart. Pyruvate kinase M2, PKM2, on the other hand, is expressed in tissues with anabolic functions and subject to complex allosteric regulations. It seems logical that cancerous cells would have preferential expression on PKM1 due to their reliance on aerobic glycolysis to produce and consume energy. However, PKM1 exists in a stable and high tetramer form with a high affinity to phosphoenolpyruvate or PEP. PKM2, on the other hand, is more commonly expressed in cancer cells and is most common in low catalytic activity dimeric form. The low catalytic activity results in an increase in glycolytic intermediates by introducing glycolyc enzymes from the pentose phosphate pathway and glycerol synthesis, resulting in the production of NADPH, thus providing materials needed for rapid cell proliferation. Sorry. According to studies, it is well known that cancerous cells grow under anaerobic conditions. Unlike PKM1, PKM2 produces minimal, if any, ATP during the last rate-limiting step of glycolysis. Therefore, under hypoxic conditions, PKM2 is activated and oxidative phosphorylation is impaired. Cancer cells get energy from mitochondrial substrate level phosphorylation via glutaminolysis. Glutaminolysis is a pathway in which glutamine, an amino acid, is converted to glutamate and then to alpha-ketoglutarate, and subsequently succinyl-CoA and succinyl for ATP synthesis in the TCA cycle. It has been well documented that cancer cells require a large amount of glutamine for cellular growth, energy, and survival. So poor Bob, I hope he makes it.